Alright, so we talked about the idea of meiosis yesterday. So today we're going to talk about some, variations of meiosis. Well, not really variations. They're still undergoing the meiosis process. We're just going to use meiosis. We're going to see how meiosis is actually really used in real life. So we're learning about oogenesis and spermatogenesis, which is the production of egg cells and sperm cells. So today we're looking purely on gametic cells because meiosis only applies for gametic cells, so basically these type. So we're gonna look at the process of a sperm cell formation. So sperm cell formation, you start producing sperm at the age of puberty, and you will continue to do this throughout life. However, the production level of sperms will lower when the age increases. This process takes place in the testes, and as you can see on the right, it slowly evolves, Golgi apparatus, all of these vesicles, and then it has this nucleus, and starting developing centrioles, develop this flagellum, and these microtubes, and it starts developing a clearer mitochondria, and then it moves this nucleus and acrosome upwards, and these excessive cytoplasm, they come down here to form this midpiece, otherwise known as a neck, and finally it becomes this full structure of the tail, flagellum, the neck, and the head. And here's the DNA material, mitochondria, and flagellum. And we're gonna talk about how sperm cells is formed. So it starts, well, everything is supposed to start, but the sperm cell starts as a diploid stem cell that's divided from mitosis when you were born, and eventually it's gonna undergo meiosis 1. So remember, meiosis 1 creates two cells. So it first undergoes meiosis 1, and it becomes two secondary spermatocytes, whatever. And then it would continue to undergo meiosis 2. And after meiosis 2, you're going to be able to get 4 spermatids. And finally, through the process of spermiogenesis, or genesis or whatever, so they will differentiate themselves, and here they were unmatured, they were just freshly produced. They're going to mature and develop into spermatozoa, other known as sperm cells. So this entire process you need to know. You don't need to know the structure, but you do need to know the structure, but you don't have to name all the organelles and stuff. So now we're going to look at specifically the sperm cell structure. The head is the nucleus, as it contains the DNA of the sperm. The midpiece, or the neck, is packed with mitochondria that provides energy for the cell movement. The tail is called flagellum, as it propels the sperm forward by whipping back and forth, like a fish, essentially. And it contains 23 chromosomes. And acrosome here, it contains some digestive enzymes. It's just there. So now we're going to look at the process of egg cell formation. So funnily enough, this process actually begins before you are born, if you're a woman, if you're a girl. So it will pause at prophase 1, meaning it will not continue from there. And it will continue when you have your puberty. When you hit your puberty and you start having your menstrual cycle. And then every egg cell produced, it will follow a menstrual cycle. And this entire process takes place in the ovaries. And as this, you can't really see it here, so I put it on the next slide. So we start off from, before you were born, 7 weeks to 4 months. So about 1,000 primary germ cells moved in developing ovaries around 7 weeks. So mitosis starts happening, and it creates millions of cells with all the baby's genetic information. So remember, we divide into all of these egg cells, all of these, primary egg cells that have not undergone any division yet. We develop all of this and put your genetic information in there, if you're a female. So this is all before birth. The cell would divide via meiosis, so that half of the genetic information is contained in each egg cell. 23, remember? So we'll have the first meiosis division ends, so the second one will start. 2 chromosomes, but half genetic information. So this is after puberty. A month, between 6 and 20 egg cells move further along in meiosis. So it will start, well, trying to divide. However, it will not unless you are, well, unless the egg cell is fertilized by the sperm cell. Yeah, so this is two different processes, which we're going to talk about a little later. So let's move on to how egg cells are formed. This is actually the important part. So at first, obviously, it came from a stem cell, and that was divided from mitosis at birth, and we talked about that. So it becomes this primary oocyte. It will stop at prophase 1 and all the way until puberty. So, wow, property. Well, okay. It will stop here and all the way until puberty. It will not grow. It will not do anything. It's just there. The secondary oocyte completes meiosis 1. However, it will not start anything after that. It will not start meiosis 2 unless it's fertilized. So we're gonna look at the weird case of what's happening with this egg cell. It has some problems. So out of all of this process and division and all of this, you only produce one actual working egg cells. This is because of the unequal cytokinesis, which then leads to unequal distribution of organelles and cytoplasm. And eventually they're dead. They're polar bodies. They don't have enough resources for them to go on and live. So the only thing that will survive is this one, and just that one egg cell. So now I want to talk more about the secondary oocyte. So if fertilized, the secondary oocyte will complete meiosis 2. It will form a fertilized zygote, other known as a ovum, and a polar body. The zygote will be implanted into your ovule, and obviously it will start developing to a baby. However, if it's not fertilized, the secondary oocyte will be degenerated, and it will be absorbed by the body. And it's hard. It's a little hard explaining without knowing grade 12. But so the corpus luteum, well, it's something a part that's in the egg. It will break down. And eventually, when that part breaks down, it will lead to a drop in testosterone levels. And when that level hits, you are going to have your menstrual period, and you're going to have your blood, and it's going to be a lot of pain. And so I taught biology before, and I had a student came up to me. He was like, "Hey, how do we have twins out of if we just have one cell? That's so weird." So I told him, there are two types of twins. There's a dizygotic twin, and there's a fraternal twin. Well, monozygotic twin. So for a dizygotic or a fraternal twin, what's happening is that there are multiple eggs released. So that's a chance. So occasionally, a woman might release more than one egg during ovulation. This can happen due to not stable hormones, genetic factors, or age, or whatever. And eventually, those eggs, if somehow, this is a very small chance that it's going to be a dizygotic twins, because you're hitting a very small chance that woman releases more than one egg that month, and you're hitting even a smaller chance that both eggs are fertilized. So this twin, when they are born, they will have their own very unique genetic setup, similar to just siblings instead of twins, and they will have different looks and all that. They're going to be looking more like a sibling than a twin. So now the common thing about twins is monozygotic, otherwise known as identical twins. So they're the identical twin because they originate from this one single fertilized cell. So how does that happen? In the early in development, the zygote will split into two separate embryos, which causes it, and this is completely spontaneous, so it will not be influenced by genetics or any other things. So both embryos, because they came from the same exact egg, they share the very same genetic material. So they will result in twins that look very identical, and they usually act pretty similar as well. So last thing is the structure of the egg cell. You don't need to know this, but it's a lot simpler than a sperm cell. You just have the cytoplasm, nucleus, and all of this corona radiata that helps. I'm pretty sure that gives support and nutrient to the egg, and zona pellucosa that helps a sperm cell to fertilize, and plasma membrane, all of it, and the rest is just membrane. It's pretty self-explanatory. Yeah, and now we're gonna look at mutations. This was the second part of today's class because we just talked about having babies. Well, egg cells and sperm cell. We just talked about them fertilizing to create a baby. We're gonna look at how possibly what's going to be this baby's genetic code. So there could be changes to the chromosome structures. So let's say we had our arrangement of A, B, C, D, E, F, G, and then suddenly maybe we undergone certain factors and it got deleted. A, B, C, D, E, F, G, or it could have gotten duplicated. A, B, C, B, C, D, E, F, G, H. Or it could be, inverted. So now everything is backward. And what's supposed to be this reading from left to right, it's not supposed to be reading from right to left. So A, D, C, B, E, F, G, H. And reciprocal, obviously, all that math stuff. So you're just going to know that there could be changes in the chromosomal arrangements. And when this change, it's usually not good. So you might be asking, hey, why does it change? So this is all because of non-disjunction. And that's supposed to be a Y here instead of that I, whatever. So it's the failure of chromosomes to actually separate properly during meiosis. So we know crossing over happens. So when crossing over happens, they're actually crossing over. They're actually crossing on top of each other. And when they do that, maybe they might not be ripped properly. Or they might not have functioned properly. And all of that, there's a bunch of problems that could happen. And human bodies, they try their best to stop that. However, chances, we have a lot of people on this earth. We can hit some really low chances. So eventually this will lead to trisomies and monosomies. So you're going to learn that. So right here, non-disjunction in meiosis 1. And that's non-disjunction in meiosis 2. So eventually you're going to see that, oh, they failed to rip apart. So all of them are in here. And none of them are in here. So now they don't have anything. So so this is only one pair, all right? I want you to understand, this is only one tetrad. So now these two, they will have 22 chromosomes. Because, we have to assume that everything else perfectly divided and everything was okay, so that only one dysfunction occurred. So we assume that these only have 22 chromosomes. So they're supposed to have 23 after now. So they didn't receive this. So now they only have 22. But these cells right here, they're supposed to have 23. All of these are supposed to have 23, including here. So, but here it got 2 from this. So now that's a 24 right there. And when we call this trisomy. And this could happen in meiosis 2, obviously. So you got past meiosis 1. Everything is okay. You're fine. They divided properly. Each cell has 2. But now, you're divided wrong. So now this becomes a euplanoid, and this also becomes a euplanoid because it has a half chromosome. Half. And now, they're the good ones. They're the good ones, obviously. They received 1, they're the good ones. They're the ones that could actually be fertilized. And these, trisomy, monosomy, bad. They cannot be fertilized. Now, what happened when you have these trisomy and monosomy? Well, that's when we gotta talk about the syndromes. So, trisomy 21 is that you have a third chromosome in the arrangement. So I actually want to show you something here. So we have all of this, 46 pairs of chromosomes. 46 chromosome set. And then we have 23 pairs of chromosome and 46 chromosome in total, and the XY chromosomes. Wait, I'm sorry. Wait a minute. So, okay. 44 chromosomes, not including 44 chromosomes, and then we have the XY. So 46 was with the XY. So you have all of these. You have 22 sets of chromosomes. They get pretty big and they get pretty small. And then you have those XYs. So XY, remember, they were determining the gender. So XX was a female. XY is a boy. So when our 21 pair of chromosome right here, instead of 2 chromosomes here, they have 3 chromosomes here now. What does that mean? That's Down syndrome. And same thing for 13. You have 3 chromosomes here instead of 2. 18, you have 3 chromosomes instead of 2. So that is Edwards syndrome and Patau syndrome. So remember, a chromosome gives you more information. So when we look at these people that I assume that most people that watch this are high schoolers. So if you're in your high school, you probably often joke about something called special ed. So these people, they're called special, and I feel like that was a very precise name because they didn't do anything bad with it. So special ed, these people, they are possibly smarter than you. They're actually no, they are smarter than you because each chromosome they're able to give us information. However, our human body is not very capable of overdriving and doing stuff. So let's say you bought an NVIDIA 4090 chip and you're at home. You're trying to get onto your PC. You're like, oh my god, I got this 4090. I gotta play something. So you started mining bitcoins and you started playing Minecraft. Or let's say you started playing Roblox at the same time. And suddenly your laptop broke down and now it can't do anything. So they're the same thing. They have overdriven themselves to have too much information in them. And now their brain is like, oh my god, what's going on? I can't process this extra chromosome. I don't know what to do with it. I don't know what happens. I know I have this one extra chromosome. I'm supposed to have more information and be smarter. But what do I do with it? I don't know. Because the human body is very not capable. So eventually these people, they just break down. They don't know anything. And now they look stupid. But they're actually really smart. And they're supposed to be really smart. And then you could actually get an extra XY chromosome. Or just one chromosome. One X chromosome, that's Turner syndrome. 3X, that's a syndrome. XYY, XXY Klinefelter syndrome. So all of this, as long as you have an extra or one, well, you can't have one less, you'll be dead. But sorry, if you have one more, then you are having some sort of disorders. However, in grade 11, we only expect you to know Down syndrome. So shoot yourself. Don't be too scared. I know you all. So as I showed this page, it was supposed to be for karyotyping. So karyotyping is a thing that we used. Well, we still use it to determine the chromosomal abnormalities. So we observe all these chromosomes. We look over. We see 1 has 2, 2 has 2, 3 has 2, 4 has 2. This is a normal kid. 1 has 2, 2 has 2, 3 has 2. This is also a normal kid. So I have a gizmo for this that you're going to do, that you're going to observe and look and see who has a disorder. But karyotyping, essentially, we're looking at somebody's DNA, we're looking at somebody's chromosomes, we're listing them out in this picture, and we're going to see, do they have a disorder or not? And that actually helped us a lot, because as if you want to become, okay, you guys are not going to, but if you're a parent, and you're at a hospital, and you want to decide, hey, do I want to have these children or not? And then suddenly doctor came out with the news. They're like, I'm sorry, they have chromosome abnormalities. They have an extra chromosome. So it will help the parents be like, oh, they're aware of it. So this is pretty beneficial. It helps us to understand, and hopefully, just hopefully, one day humans, we can establish some sort of technology that could potentially make these Down syndrome people, they are able to actually use this extra chromosome, and maybe they will become like super genius. Who knows? But yeah, that's it for this class. I hope you all do the gizmo, and next class we're gonna look at something interesting.